Rupert Sheldrake, born 1942, is a British biologist and philosopher of science whose work has been the subject of immense controversy throughout the span of his almost 60-year career. His most radical proposal, the theory of Morphic Resonance, suggests radical reconceptions of the ways in which we might understand developmental biology, animal behavior, human psychology, supposedly parapsychological phenomena, and even the nature of causation. Yet despite the potential profundity of Sheldrake's ideas, very few mainstream scientists have addressed his ideas in a serious manner, with many regarding morphic resonance with little more than dismissive scorn or outright animosity. Nevertheless, recent years have seen a notable upsurge of interest in Sheldrake's work, perhaps partially due to the excitement surrounding the work of developmental biologist Michael Levin, whose experimental and theoretical work has done much to popularize and empirically validate the concept of morphogenetic fields, largely through research regarding bioelectricity. So, with this series of videos, I would like to examine Sheldrake's theory of morphic resonance in detail, and thereby attempt to bring some clarity to questions surrounding these ideas. What exactly is the theory of morphic resonance, and what are the theory's empirical implications? Is morphic resonance a genuine scientific theory, or is it instead a form of pseudoscience? What does the theory of morphic resonance imply regarding the nature of modern science, and what relationships might there be between this theory and the philosophical realms of metaphysics and ontology? And finally, to what degree is the theory of morphic resonance empirically substantiated? In what follows, we will first be examining the theory itself, as explicated in Sheldrake's text, Morphic Resonance, the Nature of Formative Causation. We will look at the origins of Sheldrake's theory and the empirical research which led Sheldrake to develop this radically novel idea. Next, we will begin to break down the theory itself in detail, looking at empirical phenomena which Sheldrake sees as indicative of morphic resonance and the exact manner by which Sheldrake contends that such processes might occur. We will then discuss what some of the implications of Sheldrake's theory might be with regards to biology, psychology, physics, and metaphysics. Along the way, we will see the ways in which Sheldrake's ideas intersect with those of other scientists, such as biologist Michael Levin and physicist Erwin Laszlo, and with those of philosophers such as Henri Bergson and Alfred North Whitehead. Finally, we will look at Sheldrake's theory in terms of philosophy of science, examining his ideas in the light of philosophers of science such as Karl Popper, Thomas Kuhn, and Paul Feyerabend. In asking whether or not the theory of morphic resonance is indeed scientific, we will need to raise questions about the nature of science, and the manners by which scientific theories and meta-theoretical paradigms evolve over time. Alfred Rupert Sheldrake, born June 28th of 1942, began to develop an acute interest in zoology and botany in the earliest years of his adolescence. His father, a pharmacist and amateur microscopist, was very encouraging of his son's scientific enthusiasm. Much like myself, Sheldrake had, by the age of 14, renounced the religious beliefs of his family and culture in favor of a thoroughly materialistic atheism, noting that he was the only student within his Anglican boarding school to refuse to undergo the standard religious confirmation ceremony. In an interview with The Guardian, he stated, quote, "I had bought into the package deal of science equals atheism," end quote. Prior to beginning his college career, Sheldrake would work at a pharmacology research lab. Sheldrake would later recount that this experience was particularly impactful, as he was deeply unsettled by the manner in which the animals involved in the experiments conducted by the lab were required to be euthanized by the experimenters. In the years that followed, Sheldrake would go on to study biology and biochemistry at Clare College, Cambridge, later going on to study philosophy and history of science at Harvard University in the United States, and finally returning to Cambridge to receive his PhD in biochemistry with his research regarding the developmental hormones of plants. Having returned to Clare College and now with funding by the prestigious Royal Society Rosenheim Research Fellowship, Sheldrake would go on to engage in research regarding the biochemistry of plant morphogenesis. This research centered upon the role played by auxins, a form of plant hormone, in the differentiation and morphogenesis of vascular plants. Together with his research partner, Philip Rubry, Sheldrake would develop a theory of polar auxin transport, by which osmotic processes regulate the distribution of auxin hormones throughout plant tissues and thereby play a role in determining the somatic forms which such plants come to assume throughout the course of their development. Ultimately, however, Sheldrake would become disillusioned with this line of research, and with the assumptions which he and his colleagues had been making with regards to the nature of such developmental processes. Sheldrake would go on to say that the theory being developed through this research was inextricably circular, such that it was unable to actually explain differences in plant development between species. Plants with the exact same auxin transport systems nevertheless came to develop in drastically different ways, and although auxin transport processes could explain the means by which morphogenesis occurred, it was unable to explain actual differences between different morphogenetic pathways. Having abandoned his initial research program, Sheldrake would then set his sights eastward, coming to work at the University of Malaya from 1968 to 1969, and then relocating to Hyderabad, India, where he would once again devote himself to research regarding plant physiology and morphogenesis, now working with the International Crops Research Institute for the Semi-Arid Tropics. During these years, Sheldrake would also begin to take a serious interest in Indic philosophy and mysticism, studying various forms of meditation and spiritual practices. It was also during this time that Sheldrake would begin to formulate a theory which he believed might be able to resolve the problems which he initially encountered during his studies of plant morphogenesis. And it was this thinking which would eventually culminate in the theory of morphic resonance. In 1978, Sheldrake would begin to work on a book which would come to be published in 1981 as A New Science of Life, and which would later be republished, revised, and expanded to become Morphic Resonance, the Nature of Formative Causation. Within this text, Sheldrake argues that the development of biological morphologies cannot be explained simply as a result of mechanistic processes, in which organic wholes are understood as merely aggregations of smaller parts. Sheldrake proposes instead that organic physiology and behavior must be understood as being the result of a kind of resonance by which self-organizing systems are able to influence one another through formal similarity. Morphic fields, Sheldrake argues, are patterns of growth, development, and behavior which are able to inform one another across space and time, thereby giving shape to organisms in a way that transcends the supposedly determinative influence of the protein coding which results from DNA sequences within individual cells. Morphic resonance implies that DNA molecules are not, in fact, the primary units of heredity among living organisms. DNA code plays the undoubtedly consequential role of coding for protein synthesis, but Sheldrake argues that such protein synthesis is not the primary means by which the morphology of organisms is directed. Instead, organic forms are able to influence one another as such, at the level of morphogenetic fields which direct, yet do not fully determine, morphological development by influencing the behavior of the self-organizing processes which comprise living entities. Before moving on to address Sheldrake's claims pertaining to morphic resonance, I would first like to direct our attention towards the notion of morphogenetic fields more generally. Rupert Sheldrake did not himself develop the concept of morphogenetic fields within developmental biology. Rather, this idea has its roots in the early 20th century, originally being proposed in 1910 by the Russian biologist Alexander Gurevich. Although the notion of morphogenetic fields enjoyed some degree of popularity during the early decades of the 20th century, the concept suffered from a great degree of ambiguity. Oftentimes, theorists were not clear as to whether or not morphogenetic fields were intended to refer to a new type of physical field, perhaps akin to electromagnetic fields, or if the notion was simply intended to refer only to shared behavior of groups of living cells. Was the notion of a morphogenetic field best understood as referring to a new kind of phenomena, or was the term best understood as a new way of thinking about processes which were already accounted for by mainstream physics, chemistry, and biology? Sheldrake himself is quite adamant that morphogenetic fields are, as he sees them, definitively causal entities which are able to direct developmental, behavioral, and regenerative processes by means of influencing processes which are themselves indeterminate, or which we might refer to as probabilistic. This notion of morphogenetic fields came to fall heavily out of favor with the eventual rise of the neo-Darwinian synthesis, a paradigm within biology which would hold sway throughout the remainder of the 20th century and which retains a degree of dominance even to the present day. One of the fundamental assumptions of the neo-Darwinian synthesis was that organic systems were determined predominantly by sequences of DNA, which were regarded as being the basic units of heredity. With the discovery of the twisted ladder of DNA in 1953 by James Watson and Francis Crick, it was pronounced that science had finally discovered what genes were physically made of. Strings of base pairs encoded within strands of DNA coded for the synthesis of proteins, and it was concluded that this process of synthesis was the means by which characteristics were inherited and passed on by organisms, and the means by which this heredity resulted in differences of phenotypic characteristics. Natural selection would then act upon gene pools in an algorithmic manner, filtering out less fit sequences of DNA and thereby selecting for DNA sequences which express themselves as fitness-enhancing phenotypic traits. The whole history of evolution could then be understood as the creative advance of a blind watchmaker, as described by Richard Dawkins. Evolution was understood to be entirely blind and mechanical, and organisms were understood to be the machines created by this algorithmic process, entirely at the mercy of whatever sequences of DNA happen to be found within their bodies. With very few notable exceptions, such as the Baldwin effect, there was no role for agency or consciousness in directing the course of evolution, and no need for such concepts as morphogenetic fields. Morphic fields were deemed to be simply unnecessary and irrelevant, as it was declared that the mystery of life had, in essence, been effectively solved. The future of biology would simply be a matter of determining how DNA sequences came to be shaped by natural selection, and how such sequences then resulted in the physiological and behavioral characteristics of living creatures. Along with this solidification of the neo-Darwinian paradigm, the effects of the Cold War also came into play. The theory of evolution by the inheritance of acquired characteristics, originally proposed by the pre-Darwinian biologist Jean-Baptiste Lamarck, remained influential within the Soviet Union long after the theory had been abandoned by Western scientists. As such, the theory came to be ridiculed as Soviet pseudoscience by Western biologists, until eventually the field of epigenetics, through explosive developments of the field in the early 21st century, would reveal that the inheritance of acquired characteristics was indeed very real. This paradigm of neo-Darwinism remained an almost entirely unquestioned dogma until at least the first decade of the 21st century, and in many regards it remains the central paradigm of mainstream biology to this day. Nevertheless, recent developments within the field of developmental biology and epigenetics have shown this paradigm to be standing upon increasingly unstable grounds. The entire field of epigenetic inheritance calls into question many of the central dogmas of neo-Darwinism. But here we will look specifically at the work of biologist Michael Levin of Tufts University in Massachusetts, as Levin's work has come to reaffirm the veracity of morphogenetic fields, specifically bioelectric fields, as an essential concept within biology. To be immediately clear here, Michael Levin's research regarding morphogenetic fields does not involve morphic resonance per se. Sheldrake's theory of morphic resonance requires that morphogenetic fields be, in some sense, non-local, and thus able to influence one another in an indirect manner. A kind of spooky action at a distance pertaining to the inheritance of morphological influence. Levin's research itself involves no such conception of field non-locality, but it does nonetheless demonstrate that such fields do indeed exist in some manner. In particular, Levin's research has demonstrated that morphogenetic fields consisting of bioelectric gradients found throughout living cells are involved not only in the morphogenesis of individual organisms, but also in the transference of hereditary information. Much of Levin's research is centered upon experiments making use of planarian flatworms of the phylum Platyhelminthes. Planarians demonstrate extraordinary regenerative capabilities, and researchers have demonstrated that planarians are remarkably able to retain memories even after being decapitated and subsequently regenerating their entire head and rudimentary brain. This indicates that the memories of these organisms are not in fact stored within the brain, as traditionally assumed, but instead are somehow encoded within the bioelectric fields, which also allow for the regeneration of the organism following dismemberment. By means that are as yet poorly understood, the bioelectric field of such an organism is believed to be able to influence the form of the regenerating brain in such a manner that the creature's behavior continues to accord with learning which occurred prior to dismemberment and subsequent regeneration. Other experiments conducted by Levin and his colleagues have shown that bioelectric fields allow for the transmission of hereditary mutations without any alteration whatsoever to the organism's genetic makeup. Levin's team was able to alter the bioelectric fields of certain planaria, such that the organisms, rather than regenerating a missing tail, came to regenerate a second head instead. As these two-headed planaria came to reproduce, the offspring also generated two heads rather than a tail. All of this was achieved with no alteration to the DNA sequences of the organisms, but rather by the manipulation of morphogenetic fields. Levin and his team have also conducted experimental research upon developing tadpole embryos, with equally insightful results. In one experiment, Levin's research team rearranged the developing organs of their frog embryos in random ways. If these organs continued to develop from arrangements which the researchers had artificially imposed, then these embryos would have resulted in the development of horrifically disfigured adult animals, with none of the mature organs arranged in proper relation to one another. Instead, Levin found that these embryonic organs were able to relocate themselves, moving different distances and in different directions, such that the embryos eventually came to develop into perfectly normal adult frogs. The embryonic organs somehow knew that they were not properly arranged in relation to one another, and were able to relocate themselves as needed in order to compensate for this. Levin has argued that such phenomena are the result of gradients of stress, or unrest, between cells, which are determined by the form of the bioelectric fields which give shape to the organism. When the arrangements of cells and tissues accords with that prescribed by the bioelectric field, the cells experience very low unrest and thus proceed to carry on their normal functioning. When there is a disconnect between the arrangement of the cells and the bioelectric field, the cells become stressed in such a manner that they then relocate, reproduce, self-destruct, or differentiate until their stress levels reach a resting state and the correct form of the organism is thereby instantiated. It is thus bioelectric information, the information of morphogenetic fields, which determines the physiological development of the organism at the highest levels. As Levin has stated, it seems to be that the role of DNA is not directly determinative of an organism's overall morphology, but rather that the DNA instead determines the lowest level features of individual cells. DNA determines the hardware, as it were, while the morphogenetic fields determine the higher level morphology of the organisms. This morphogenetic research, although far from a validation of Sheldrake's theory of morphic resonance in and of themselves, spell very serious trouble for the neo-Darwinian synthesis for a number of reasons. It has been a central shibboleth of the neo-Darwinian paradigm since its inception that phenotypic traits and hereditary transfer were mediated primarily, if not exclusively, by the information contained within strands of DNA. Units of heredity were dubbed genes, and genes were identified as being sequences of DNA. If heredity, including enormous mutations such as two-headedness, can be passed on without any alteration of DNA, then it must be conceded that genes must also be comprised of the characteristics of bioelectric fields. If a zany scientist in a lab can produce such a massive mutation via manipulation of such fields, then it seems almost assured that such fields may come to alter themselves throughout the course of an organism's lifetime. Alterations which, with no mutations of DNA strands required, would then be passed on to subsequent generations of such organisms. The potential implications of such findings are vast, and unfortunately extend far beyond the scope of my project here. For example, we have, for over half a century, regarded genetic as synonymous with immutable characteristics we received without our consent from our parents and ancestors. If, alternatively, our genetic inheritance consists of morphogenetic fields which we might be able to alter through our behavior or environment, and which we might then pass on to our children, then the implications for the sciences of health and medicine could be immense. Levin's research might also suggest that the influence of macromutations upon the history of evolution might be far more significant than what has generally been assumed by the neo-Darwinian clergy. Throughout numerous books and essays written during the 1980s and 90s, the American biologist Stephen Jay Gould came under intense scrutiny for his theory of punctuated equilibrium. Gould argued that the evolution of life on Earth was mostly characterized by long periods of relative evolutionary stability, punctuated by periods of rapid speciation and development, which often correlated with ecological and geological catastrophes. Gould was thoroughly criticized and even ridiculed by other evolutionary theorists, most notably Richard Dawkins and Daniel Dennett, who claimed that Gould's theory was untenable, as such rapid jumps forward in evolution would require highly improbable macro-mutations. Gould's critics insisted that Darwinian evolution was necessarily a gradualistic process, by which only tiny mutations compound upon one another over the course of millions or billions of years, so as to eventually result in large evolutionary transformations. The macromutations implied by punctuated equilibrium could not play a significant role in evolution, so it was said, as such mutations would result in the characteristics of an organism being very far removed from the hill of optimal fitness which its ancestors had been climbing within the fitness landscape. Thus, only micromutations ever had any real chance of being beneficial to an organism, and therefore evolution must be a necessarily gradualistic process without any punctuating leaps. This argument appears rather solid if we are assuming that phenotypic characteristics are the direct and immutable result of DNA sequences. Randomly making tiny alterations to the code of your computer program has a very low, yet nevertheless real, probability of positively impacting the performance of your software. If you iterate this algorithmic process for millions of years, being sure to keep the positive alterations while throwing out the negative changes, then evolution will indeed occur. Alternatively, if you are randomly making very large alterations to your code, it is very unlikely that you will ever get anywhere, as each mutation has such a vanishingly small chance of being a positive alteration that the selection process will not be able to culminate into a net positive direction. However, if we regard organisms as being truly organic, as being self-organizing systems rather than being analogous to computational machines, then it may be that the reality is quite different. A two-headed planarian might not be a great example of a viable macromutation, but two-headed planarians are nevertheless able to survive and reproduce despite being drastically and artificially altered by experimental methods. The organism's tissues and organs are still able to function because they know what they are trying to do, and are smart enough on their own to figure out how to accomplish their goals without those goals being explicitly coded in, as is the case for computer programs. It isn't difficult to imagine situations in which a macromutation such as the duplication of a body segment, organ structure, appendage, or cluster of neurons might originate as a neutral or even slightly harmful alteration, which then comes to be adapted purposefully by the organism so as to perform tasks which were simply not possible prior to the mutation. The history of evolution on Earth may prove to have been a punctuated equilibrium after all. Further, if morphic fields are, as Sheldrake and Levin both suggest, also in some sense cognitive, then understanding the process of evolution as the purposeless, mechanical advance of algorithmic selection processes may be a hopelessly inadequate framing of life's history on our planet. We might instead see evolution as the feeling forward of distributed, collective consciousness which is perennially drawn upwards towards new heights of novelty, possibility, and numinosity. Life might be better understood as a dreaming gardener, rather than the blind watchmaker suggested by Richard Dawkins. There is much more to say regarding Michael Levin's research and its implications, and I would highly recommend that anyone interested in these topics check out his channel here on YouTube, to which I'll be adding a link in the description along with a link to Rupert Sheldrake's channel. For now, however, the major takeaway is that, just as Sheldrake surmised back in the early 80s, the processes which give shape to organic morphology are top-down, as it were, more so than they are bottom-up. Put another way, we could say that the software of life which is encoded and perpetuated within the actual morphology of organisms is at least as important as the nature of the individual building blocks which are synthesized with the help of DNA. Before moving forward, I should also mention that there may not be simply a one-to-one identity between the morphic fields proposed by Sheldrake and the bioelectric fields studied by Michael Levin. It could be that bioelectric fields are only a special case of a more general phenomena of morphogenetic fields. Some may be comprised of bioelectricity, while others might be comprised of chemical systems or other such processes which are as yet unidentified by contemporary science. Alternatively, it could be eventually discovered that bioelectric fields do indeed exhibit the properties which Sheldrake has theorized. As for now, these remain open questions. Nonetheless, developments in research pertaining to bioelectric fields has certainly shown that Sheldrake's intuitions pertaining to morphogenetic fields were well-founded, despite how much his thinking may have been misaligned with the scientific zeitgeist of the 20th century. Yet the most significant and radical aspect of Sheldrake's theory is not merely the effects of morphogenetic fields upon development, heredity, or behavior, but rather the effects of what he refers to as a resonance between morphic fields, a much more radical proposal with much more radical implications. Sheldrake contends that morphic fields do not simply transfer in a linear fashion from parents to offspring, but further that morphic fields are able to influence one another non-locally and even across expanses of time on the basis of morphic similarity. The most basic form of resonance which Sheldrake describes is self-resonance, by which an organism's morphic fields are influenced by their own past states. Morphic fields naturally alter themselves throughout their developmental cycles, thereby facilitating phenomena such as embryonic development and metamorphosis. Yet, morphic fields, due to self-resonance, also exhibit a kind of inertia, as a field attempts to maintain its own form over time. Consideration of such phenomena could potentially help us to understand the nature of addiction, ritualistic behaviors, and many forms of neurodivergency among human beings, such as obsessive-compulsive tendencies. Sheldrake contends, however, that the effects of such resonance extend far beyond the effects of an individual's past upon the present. Sheldrake argues that in order to fully understand the nature of living systems, we must also realize that the influence of morphic fields is able to occur between individuals, thereby resulting in a form of automatic averaging, by which self-organizing systems such as bodies, behavioral patterns, and learning dispositions are able to inform one another in a horizontal manner. In the next episode, we'll be looking at specific lines of evidence which Sheldrake puts forward in order to illustrate his theory, and empirical evidence suggesting that resonance between morphic fields is indeed a phenomena which can be observed. Along the way, we will be examining the theory itself in greater detail, and illustrating the exact nature of the process of morphic resonance as Sheldrake envisions it. We will then consider morphic resonance in ontological terms, considering what morphic resonance might imply regarding the nature of time, causation, and even the nature of the laws of physics. But that's going to be it for this episode. Do me a solid and like and subscribe if you want to see more videos like this. Be sure to drop a comment if you have any thoughts or questions. And thank you for watching.